Biology Part 88, and the two processes are, we will explain them over here. Chondric ties, and the cartilaginous fish such as sharks and dogfish are able to tolerate concentration of urea in the blood 150 times greater than those found in the blood of other vertebrates. This creates an artificially high blood osmotic concentration and may reduce the osmotic gradient across the gills to zero. Apart from the usual degree of urea reabsorption, their kidney function is similar to that of the mammals. Number 2. Osteichthyes, bony fish like cod and herring replace water lost through the gills by drinking seawater and consequently requiring mechanisms to cope with an excessive salt intake. By far the most important of these mechanisms is the active excretion of the excess salt at the gills, a reversal of the salt pump possessed by the freshwater bony fish. The kidneys of the marine bony fish have a very small fluid output and usually lack glomeruli. Waste products are selectively extracted from the blood by the tubule cells and ultrafiltration does not occur. One further consequence of the physiological drought is the inability to excrete nitrogenous waste as ammonia, which being toxic needs to be diluted by large quantities of water for expulsion. To avoid this, ammonia is converted into non-toxic substances, trimethylamine oxide, which is removed by the kidneys. Migratory bony fish such as seals and salmon can change physiologically from salt water to the fresh water back again, and this ability is poorly understood but implies the possession of the two alternative sets of genes controlling osmoregulation. Desert mammals. The desert mammals show a remarkable ability to control water loss in dry environment. The camel, for example, has a number of typical and few rarer physiological modifications for desert life. It can eat dry food, drink water equivalent to 33% of its body weight, and at one session, absorbing the water only slowly from its alimentary canal to avoid dilution of the blood. It should be noted that the chemical process of the respiration produces significant amounts of water. In the camel, this respiratory water supplements dietary intake to such an extent in the winter when fresh green plants are available. The animal has no need to drink water, while in summer it only drinks infrequently. The camel is humped in a store of fat and which can be metabolized to provide water in times of shortage. A further important modification common to many dry desert concerns with the loops of Henle and collecting the ducts of the kidney tubules. These are much longer than in the other animals, enabling a very concentrated urine to be formed. Although primarily an adaptation of the conserved water, this mechanism also increases the animal's ability to excrete salt so that the camel can drink salt water up to 75% of the seawater. And as in the other ruminants, the need for nitrogenous excretion is reduced by the secretion of urea into alimentary canal. Bacteria in rumen converts the urea into protein, which is then utilized as food. Flowering plants, in common with all the land organisms, terrestrial flowering plants must conserve water in order to survive. Water is lost through the leaves during gas exchange and has a continual flow of water that must be maintained to meet the plant's needs for transportation and structural support, of course. In general, plants can control the amount of the water lost by opening and closing the stomata according to the external conditions. In case where an excess of water accumulates in a plant, special water expelling glands called hydatodes described earlier may occur. During the periods of drought, the plant may cease activity or die, leaving resistant seeds which germinate when favorable conditions return. Physiological drought occurs when the ground is frozen.